During meiotic division, two things happen. First, one diploid nucleus divides by meiosis to produce 4 haploid nuclei. And second, the separation of pairs of homologous chromosomes in the first meiotic division creates half the chromosome number. The process of meiosis consists of 2 cellular divisions, the first meiotic division and the second meiotic division. During the first meiotic division, the separation of pairs of homologous chromosomes creates half the chromosome number, making it a diploid number to a haploid number. The second meiotic division separates sister chromatids, which are created by the replication of DNA during interphase.
Meiosis consists of 2 divisions, both of which follow the same stages as mitosis: prophase, metaphase, anaphase, and telophase. Meiosis is preceded by interphase, in which DNA is replicated to produce chromosomes consisting of 2 sister chromatids. A second growth phase called interkinesis may occur between meiosis 1 and 2. However, no DNA replication occurs in this stage.
The first meiotic division is when the diploid cell becomes a haploid cell. It is a reduction division in which homologous chromosomes are separated. The first stage of the meiotic division is prophase 1. This is where the chromosomes condense, the nuclear membrane dissolves, homologous chromosomes form bivalents, and crossing over occurs. The next stage is metaphase 1. This is where the spindle fibers from opposing centrosomes connect to bivalents at centromeres and align them along the middle of the cell. The next stage is anaphase 1. At this point, the spindle fibers contract and split the bivalent, causing the homologous chromosomes to move to opposite poles of the cell. The last stage of meiosis 1 is telophase 1. This is where the chromosomes decondense, the nuclear membrane may reform, and the cell divides to form 2 haploid daughter cells. The last part is called cytokinesis.
Meiosis 2. This second division stage separates sister chromatids. These chromatids may not be identical due to crossing over in prophase 1. Prophase 2. This is where chromosomes condense, the nuclear membrane dissolves, and centrosomes move to opposite poles. The next stage is metaphase 2, which is where spindle fibers from opposing centrosomes attach to chromosomes at a centromere and align them along the cell equator. After this stage is anaphase 2. In this stage, spindle fibers contract and separate the sister chromatids, and then the chromatids, which are now called chromosomes, move to opposite poles. Telophase 2 follows, and this is when chromosomes decondense, the nuclear membrane reforms, and cells divide through a process called cytokinesis to form 4 haploid daughter cells. The final outcome of meiosis is the production of 4 haploid daughter cells. These cells may all be genetically distinct if crossing over occurs in prophase 1, which causes recombination of sister chromatids.